Biology 1010, presentation number 10, Biotechnology. Genetic engineering. We've all heard of it. It's sometimes also referred to as recombinant DNA. Sometimes it's referred to as gene technology. No matter what the title, it is a process of inserting novel or new genes into an existing organism to achieve some desired result. That organism could be a bacterium, it could be a plant, it could be an animal. But by inserting those genes into that organism, you might confer disease resistance, improved growth, improved productivity, desirable function, improved quality of product. You might be able to make known substances and, in some cases, actually make new or novel substances that have not ever been made before, but might have applications. Many of us have come to think of genetic engineering as a human-based process. I mean, after all, humans genetically engineer things. On the other hand, we need to understand that genetic engineering can be a natural process. As a matter of fact, originated as a natural process probably billions of years ago. So let's take a look at what qualifies as this business of recombinant DNA as being a natural process. Now think about it. If we use the term recombinant DNA, it means we're making new DNA combinations. On our slide, we see our first bullet point tells us that the natural process of recombinant DNA, or the natural process of genetic engineering, can be the transformation of bacteria. And our understanding is this process occurred long before humans were ever on the earth. Case in point, you have a bacterium on the left, and it dies. When a bacterium dies, it may in fact break apart, open up, and release its molecular contents into the environment around it. Those molecular contents include fragments of DNA. The DNA contains the genes that made that bacterium what it was, how it was, and how it behaved. You've all of a sudden released these fragments of molecules into the environment. The bacterium's dead, but the genes are still there. You've got some other bacterium present, at which point the fragments are taken up by that bacterium. You see the bacterium there with the blue DNA molecule in it. It's alive and kicking. And what happens is that fragment of DNA becomes integrated into this living bacterium. Now, as I look at that red piece of DNA could represent genes that bacterium never, ever, ever had before. That means that bacterium has been genetically engineered. That bacterium has recombinant DNA, and it's all been done in the absence of human intervention. It can be a natural process. But that's not the only way it can happen. My second bullet tells me about something called plasmid transfer. Bacterial cells a lot of times will have these little round circles of molecules called plasmids. You will become familiar with them as you work on the OpenStax project that's required in this course. But if you take a look at the center illustration on the right-hand side of the slide, you will see a series of pictures showing you a bacterium on the left that has a plasmid, and that plasmid is illustrated in red. The bacterium has its own DNA molecule, which is illustrated in green. The plasmid is just an teensy-weensy, teeny-tiny little additional molecule of DNA that can be transferred. And so you will see that between the bacterial cell on the left with the plasmid and the bacterial cell on the right without the plasmid, we are going to form something called a conjugation bridge. It's indicated by the arrow. Now, unfortunately, the word conjugation implies sex, but in this case, it is nothing more than a cytoplasmic connection between two bacterial cells. There's no sex going on here, but what's going to happen is that plasmid found in the bacterium on the left is going to replicate, and one of them is going to move into the cell on the right. And as soon as the cell on the right receives the plasmid from the bacterium on the left, the cell on the right now has become transformed. It has recombinant DNA. It is genetically engineered. And once again, it has happened in the absence of human intervention. There's a third way this can happen. Third bullet talks about viruses. There are viruses that infect bacteria, believe it or not. So the viruses that infect bacteria are called bacteriophages. And a bacteriophage can act as a vector. Now, the word vector means carrier. So what we can do is have a virus land on a bacterial cell, much like the artist's illustration at the bottom left. What looks like an old-timey lamppost is a virus. It's a bacteriophage, and it is landing on the surface of a bacterium like the other bacteria that you see in the background in the picture. That virus will inject its DNA into the bacterium, at which point the bacterium will be held hostage, and the bacterium will crank out lots more viruses, as shown on the right-hand side of the illustration. But the fact of the matter is that virus, by virtue of injecting its DNA into the bacterium, could conceivably impart its DNA into that bacterium. It might be a gene that the bacterium has never had nor seen before. And it is quite possible that if that bacterium does survive the viral infection, it may well appropriate that particular gene and do something it never had done before. So genetic engineering is something that could happen in the absence of humans. It has been happening in the absence of humans, and it's been critical in the transformation of bacteria over literally billions of years. Now, the word transformation is a common one that is used to describe this idea of recombinant DNA. Transformation means just simply genetically transforming an organism by adding a new gene. Now, it's not just limited to bacteria. The next major bullet on this slide tells me it can happen in eukaryotes, which would be the cells of plants, which would be the cells of animals, could be the cells of fungi, could be the cells of protists living in an ocean or a pond somewhere. Bacteria commonly infect eukaryotes, so bacteria could be vectors of these genes. Viruses could be vectors of these genes, and parasites could be vectors of these genes. The bottom line is if a new gene can find its way into a bacterium, a new gene can certainly find its way into a eukaryote. And it is and has been a natural process. It just happens that human beings figured out the process about 40 years ago, maybe, and then actually moved it into an application where we actually can do genetic engineering in the laboratory. But things have been going on in the natural world for millions and millions and millions and hundreds of millions of years. Okay, so human beings can do this. And that, of course, took the discovery of DNA structure way back in 1953 by Watson and Crick, and a whole lot of scientific research since that time. So human beings can genetically engineer bacteria, we can genetically engineer plants, we can genetically engineer animals, and we pat ourselves on the back because we are so intelligent we can do these things. But in order to do these things, we need some tools. So our tools are going to be discussed on the next series of slides. One of the very first tools or procedures we need in the process of genetically engineering organisms is DNA extraction. If I am going to modify DNA, and it doesn't matter whether it's a bacterium or an animal or a plant, I need to be able to extract that DNA. So we're going to take a look at strawberries here, and we're going to very quickly run down the steps that are necessary to extract DNA. And it's important because, number one, we need to remember where DNA is located in the cell. We'll need to understand that we need some method of getting it out. And then we need some method of being able to manage it so we can do something with it. So here's the steps. Number one, break the cells. That's a process called lysis. And we're going to use a substance called a detergent, which is like a soap, in doing that. Adding detergent to a bunch of strawberries and putting them in a blender will disrupt the cells and will disrupt the nuclei so that the DNA is freed. But now we also remember something from our presentation on DNA way back in the days, that DNA is packaged along with protein. Well, frankly, protein is going to get in the way. So step number two, we need to unpackage the DNA from the protein that holds it all together. And we're going to use an enzyme called a protease. Protease, think P-R-O-T means protein. ACE is an enzyme that breaks that stuff. So step one, break the cells with a detergent. Step two, unpackage the DNA with a protease. Step three, the only step we have left to do, is to take this DNA substance, which at the present is soluble or dissolved, and we need to convert it into an insoluble or undissolved form. If you take a look at the test tube at right, you will see a red zone at the bottom of the test tube, which is this strawberry juice that would include dissolved DNA. And in the clear section immediately above is a layer of ethanol, which is alcohol. Very simply, what happens if DNA in its soluble form comes into contact with ethanol, it will then precipitate or become solid. And looking at that test tube, you'll see what looks like some goopy mucus-like strands in that clear area, and those are DNA filaments, parts of the DNA molecule. So what we've done is we extracted DNA from the strawberry cells. We've extracted the DNA from the proteins that bind it, and now what we've done is converted that dissolved DNA into an actual solid form. So I strongly suggest that you make sure you know these 3 steps. So DNA extraction is our first tool. What comes next? Well, we'll talk about something called the polymerase chain reaction. It is sometimes abbreviated PCR, but I certainly would prefer you to remember it as polymerase chain reaction. Now, the polymerase chain reaction is absolutely necessary because in many cases, when we extract DNA from something, there isn't an awful lot of it to work with. The problem is you might extract DNA and you get this teeny tiny little microgram amount of DNA, and it's not enough for you to work with. Somehow we need to be able to create copies of that and amplify it, make more of it. So if I'm starting with a microgram of DNA, well, maybe I can get a full gram or even a half a gram, and that might make it worth my while. So this thing called the polymerase chain reaction is a process where you start with one teeny tiny sample of DNA. You'll see it illustrated in the picture there on the left. And through an automated process, you make 2 copies, make 4 copies, make 8 copies, and so on and so forth. Polymerase chain reaction is going to involve an enzyme called a polymerase. It makes a long chain molecule. DNA is a long chain molecule, and all we're going to do is we're going to make copies over and over and over again. So the polymerase chain reaction is a process by which we make more DNA of what we start with. Sometimes we'll want to cut the DNA in order to open it so that we can insert a new gene, or sometimes we might want to cut above a certain gene and below a certain gene in order to remove it from a DNA strand. Regardless, there are enzymes that function like little snips or like little scissors that will do this job for us. Once again, this is something you are going to become familiar with as you do the OpenStax project. These enzymes are called restriction enzymes, and the word restriction indicates, at least to me, that they're going to be very restricted in terms of where and how they cut. In this illustration, you will see a strand of DNA, and in the center of that strand of DNA, you will see an area that's highlighted. That area that highlighted there is specifically cut by that restriction enzyme called EcoRe that you see illustrated smack dab in the middle. It's that big oval blue thing. And that restriction enzyme, sometimes called a restriction endonuclease because it cuts within the strand of DNA, is going to cut in a very specific place. And you see the cut it makes is an uneven cut. It's kind of a zigzaggy cut. And a zigzaggy cut results in the formation of what are commonly referred to as sticky ends. And those sticky ends give us the capability of being able to splice these molecules to other molecules later on. All you have to do is have the same restriction enzyme make the cut. And because those two cuts are identical, it's going to be very easy to splice one end to another end because all of those little A's, C's, T's, and G's are going to match up perfectly. Occasionally, scientists have to get creative about getting DNA into cells. And one method they use is called electroporation. And it's a process where you use electricity to create a hole in a cell to facilitate gene transfer. You might remember something about membrane structure. It's nothing more than a bilayer of phospholipid molecules with occasional protein. The illustration on the left shows me a typical bilayered arrangement of phospholipid molecules in a membrane. There's a little itty-bitty piece of DNA I want to get into the cell. It's not getting there because the membrane's in the way. So what I'm going to do is I'm going to go ahead and apply a very small electrical pulse. That electrical pulse is a little itty-bitty bit of electricity that will disrupt the structure of the membrane in just a minor moment, enough for that particular piece of DNA to slip from the outside of the cell into the inside of the cell. It's a means by which we can get genetically modified DNA into a cell. We have already mentioned using plasmids as vectors. As a matter of fact, we're going to use plasmid as a vector in doing the OpenStax project, so we're not going to dwell a tremendous amount of time on this particular slide, just understanding that within these bacteria are these small circles of DNA that can be opened. All I need is a restriction enzyme. I open the circle. I have a piece of gene that I want to insert in that circle. And as long as I use the same restriction enzyme to cut the gene out, the sticky ends are going to match perfectly. And as a consequence, I have a genetically engineered plasmid on the right there. Plasmid in green, new gene in red. I can insert that into a bacterium, and all of a sudden, that bacterium has a gene it never had before. All I need to do is grow that bacterium. It will divide very, very rapidly, and it will make anything that that gene tells it to. Not content to just shift new genes into new cells and new organisms, scientists have been really interested in being able to actually look at the sequence of the gene to figure out what makes it different maybe from other genes. You will remember that DNA sequences consist of little letters, A's, C's, T's, and G's. And those letters, remember, are grouped in sets of 3, or triplets. And we remember that each triplet might code for start making a protein or end making a protein, or could be the amino acids that make the protein up to begin with. So you run down the length of the gene and you're reading what literally is a recipe for making a new protein. So scientists want to see what the recipe looks like. And that's what DNA sequencing is all about. It's looking at the gene in a much more close way. And so what we're doing is we're going to not just understand the gene makes a protein, we want to know exactly what the letters are in the sequence that result in that protein. So I'm looking at this picture on this slide and I see all these A's, C's, G's, and T's, and that literally is an actual sequence taken from a particular gene. They look like that. And I know it seems terribly, terribly boring, but it's so terribly, terribly critical because if you know the specific nucleotide sequence, you know the specific amino acids that make up that protein. And so if you want to modify the protein, modify the sequence. If you want to know why a protein, for example, in a cold water fish is different from protein found in a warm water fish, obviously the proteins might be critical to their survival in the environment which they live. Well, what you could do is you could look at the basic instructions for making those proteins, and you might be able to make some conclusions about the relationship between the fish, either on a physiological sense or even at an evolutionary sense. One of the more interesting aspects of our understanding of genes and gene sequences and proteins is the idea that we can do something called DNA fingerprinting. Most of us have heard about DNA fingerprinting watching things on TV or reading books or novels about crimes and suspects and evidence and things like that. So the basis of this particular procedure, we're going to call DNA fingerprinting, begins with the idea that you and I have DNA that is composed of sequences that are quite different from each other. My DNA is different from yours. Our DNA molecules are unique because they contain unique sequences. If we can break our DNA into fragments, we can take those fragments and we can migrate them through an electric current. The fragments move based on how much they weigh and what their relatively charges are. And so you'll see on this picture on the right-hand side a series of bands that have stripes in them. And those stripes represent DNA fragments that have moved a particular distance. And so you'll notice I got a pattern of fragments shown in the column called crime scene. And I've got similar patterns in columns labeled suspect number 1, suspect number 2, and suspect number 3. Now, I'm interested in determining who is guilty here. So let's suppose I've taken a DNA sample at a crime scene. The DNA sample at a crime scene gives me the DNA pattern in column 1. It matches up perfectly with suspect in number 2. So if I really wanted to make a connection between a suspect and a crime, I would make the connection between the DNA obtained at the crime scene with the DNA of the suspect. And you notice that the patterns are different for suspect number 1 and for suspect number 3. At which point, even though they might seem to be guilty at first hand, they're probably not. But suspect number 2 with the perfect alibi is linked to the scene of the crime by his or her DNA. So DNA fragments migrate through an electric current, creating a unique pattern, and it's going to use a process known as electrophoresis, which we're going to see on the next slide. So what is this electrophoresis all about? Well, electrophoresis would imply that we're going to have some kind of electric field. So what I look at in this illustration at the right is this big thing that looks like an ice skating rink. It is a big box that contains a gel. And that gel, which is a gelatinous-like material, is a solidified material called agarose. You will notice on the left-hand side of the gel is a negative cathode. On the right-hand side is a positive anode. In other words, those are connected to a power source. So you turn the power source on, I'm going to get an electric field between the negative end and the positive end. Now you'll notice on the negative end, I've got a series of what looks like holes in the gel. And those holes are places where I can put mixtures of DNA molecules. They might be obtained from a crime scene. They might represent suspect number 1. They might represent suspect number 2. Regardless, those DNA fragments are unique, and all DNA fragments have a negative charge on them. So that when you turn on the power, the negatively charged DNA fragments are going to automatically migrate through that gel from their location on the left towards the positive pole on the right. And that occurs in the electric field, and in the illustration at the bottom, you see that those fragments kind of drop out, and they form this pattern. The basic idea is that if a fragment is really, really big, it doesn't move very fast. If a fragment is really, really small, it moves a lot faster. And so the bands indicate how these fragments kind of sort out as they move. And obviously, my fragments are going to be different from yours, and your fragments are going to be different from anybody else's. And so the pattern of fragment migration we get indicates the differences and similarities if they do exist. All right, so at this point in the presentation, we have covered all the tools. We understand that genetic engineering originally started as a natural process. Human beings kind of co-opted it when we learned about DNA. And then we've come up with a bunch of tools that allow us to do the job. But now what we're going to do is take a look at the results. When human beings do this genetic engineering thing, we create what are called transgenic organisms. A transgenic organism technically is a transformed organism. They're genetically modified organisms, sometimes called GMOs. You've heard of them before. And any GMO is an organism that's had its genes manipulated. Now, sometimes those genes are manipulated to improve product quality. So you might get a better strain of corn or a better strain of tomato. It might improve resistance to pests and disease. I could genetically modify strawberries. I could improve resistance to herbicides. I could genetically modify soybeans. I could confer novel abilities on the organism. For example, I could create a transgenic bacterium that collects gold, that is elemental gold, from material within which it's living. And it's possible. Now, sometimes I can actually generate a transgenic organism to crank out marketable substances. For example, if you could go ahead and Google something called, you could Google web spinning goats. It's kind of a weird situation where scientists have managed to put the genes for spider silk into a goat. And the goat creates milk, and you milk the goat, and the goat's milk contains this protein that it's used to make spider silk. So now we've all learned at some point that spider silk is tremendously strong. And you realize if we could take spider silk in large quantities, we could make cables and ropes that were extremely strong, much stronger than the ones we have today. And so by putting this gene into a goat, goats produce the protein in milk, and all you need to do is milk the goat and then treat the milk to convert that protein into an insoluble form, and you get the same protein found in the spider silk. That's an example of a marketable substance. So now we're going to run through a bunch of examples, and you're more than welcome to Google each of these, but one way we can basically generate some desired result is to improve product quality. There is a genetically modified form of rice called golden rice, and golden rice has been genetically modified to increase its vitamin A content. It's important because there are a lot of indigenous peoples in this world that don't get enough vitamin A. As a matter of fact, vitamin A deficiency is the number one cause of non-accidental blindness in the world. If you could somehow boost the vitamin A content in an individual's diet, you might not have that problem. Thing is these people tend to eat a lot more starchy things than they do proteins. And a lot of times the starchy things they eat aren't terribly high in vitamin A. You could genetically modify rice to be that way. You might be able to improve that person's diet. So golden rice is an example of where we could transgenically improve product quality. You look at Granny Smith apples over there. You and I both know that when you slice apples, it doesn't take long for the apple to turn brown. And there's nothing worse than looking at an apple that is all nasty and brown looking. But the thing is, you got to understand that a brown apple does not mean anything's wrong with it. The apple contains some enzymes that when the apple is sliced, they will automatically turn the flesh brown. Still tastes like an apple. It might not be as soft or as crunchy as the apple was when it was fresh, but it does not indicate any kind of bacterial decomposition or anything like that. But it does decrease the quality of the apple. So if you could somehow genetically modify a Granny Smith apple to decrease the amount of brown formation when the apple is cut, you're probably going to make a bunch of money. And that's another example of improving product quality. Let's take a look at improving resistance to pests and disease. That big, nasty-looking caterpillar on the right. Some people call it a tomato hornworm. Some people call it a tobacco hornworm. The bottom line is that thing is a voracious pest. I have seen those things in my garden, and they can devour a tomato plant overnight. So it's something I don't want. So what I could do is I could build resistance into my tomato plants, into my corn plants, into my rice plants by using a gene from a bacterium called Bacillus thuringiensis. Now I know that sounds kind of obscure, but the gene in Bacillus thuringiensis codes for a toxin that gives that insect a fatal tummy ache. So if that insect crawls up on that corn plant or that tobacco plant or that tomato plant and starts eating it, it will get a stomach ache that will end its life because that plant contains the exact same toxin found in that bacterium called Bacillus thuringiensis. Now, immediately below the picture of the caterpillar, there is a picture of a papaya plant. Papayas are a tropical fruit, at least a subtropical fruit, and the DNA found in papayas these days confers some degree of resistance to a virus called papaya ring spot virus that has been detrimental to the crops produced by these tropical and subtropical countries. And arguably, that's a good thing. If your country is not terribly wealthy and your farmers are not terribly wealthy, it would be really, really nice if you could actually decrease the level of plant disease and increase the level of productivity. So you might get a bigger domestic product and they might get more money. Now, sometimes a transgenic organism might seem to make sense until you think about it in terms of the larger picture. What we see here is a picture of, obviously, a farmer holding a bunch of soybeans, and I see a field of soybeans. Soybeans do not do well when there are weeds around, and herbicides are substances that are really good at killing weeds. One of the more efficient herbicides is something called glyphosate. Problem is that glyphosate will actually kill a soybean also. So there was a reasoning amongst some scientists in, I believe, a major organization called Monsanto. And so they decided, well, what we could do is we could create a transgenic soybean. And if we create a transgenic soybean that's resistant to glyphosate, we could spray glyphosate all over the field, kill the weeds, keep the soybeans, and improve our crop. Now, that sounds like a great idea, but think of this. Fact of the matter is, we already know that genes can move from organism to organism, which means a gene for resistance to an herbicide might not necessarily stay in that soybean. It might wind up going into one of those weeds that we want to kill. So all of a sudden, by creating a transgenic soybean that's resistant to this herbicide in order to boost soybean production, we may actually create a whole brand new set of weeds that are resistant to that herbicide, which literally means we have to find a new herbicide to kill those organisms, which means then we'd have a form of soybean that is designed to be resistant to herbicide that we can't use anymore. And these repercussions can have an effect on all other crops, all other plants. You can see that it could become a very, very thorny issue. Even though we started off meaning well, the ultimate result could be a complete and total mess. With the whole notion of climate change, scientists have come to realize that there's probably going to be a global shift in crop distributions. So therefore, farmers who ordinarily grow corn, for example, in a particular area might be dealing with drought or hotter conditions as the climate changes. So if we could create a transgenic variety of corn that might be a little bit more resistant to drought, that might be a good thing. So a company called Monsanto is now promoting a genetically modified variety of corn called drought guard corn for that very same reason. I had already mentioned the web spinning goat as an example of a way to generate a marketable substance, and so this particular slide documents that. And once again, you could go ahead and Google web spinning goats. The product I had understood was called BioSteel, but in a Google search, that wasn't giving me any results, although I didn't really look very carefully. But the fact of the matter is, if you could insert that gene into the goat, you could harvest all this goat milk and have this protein ready to create any type of cable or rope or fiber you'd need to. And it would certainly be a much bigger diameter than a spider thread, and therefore result in a tremendous amount of strength that you wouldn't ordinarily find in a rope made of hemp or nylon or even a cable made of steel. Pollution remediation is another application of transgenic organisms. Scientists have been able to genetically modify some bacteria so they're actually able to metabolize oil. And so if you had an oil spill, instead of washing the rocks off with hot water, which they did once upon a time, you could basically spray a suspension of these bacteria on those rocks, and the bacteria would be quite happy to eat up the oil so it wouldn't necessarily get back in the ocean or coat birds and kill fishes and all that sort of thing. In the context of another slide, I do remember genetically modifying bacteria to perhaps extract gold from the material in which those bacteria are living. Gold is a pretty darn valuable thing, and at this point, a lot of the gold that we find in this world is distributed in such a diffuse manner that it's really impossible to get it in sufficient quantities to make it worth our while. When I go out and mine gold, I find a gold vein or a high concentration of gold, and at that point, it becomes worth my while. However, I could essentially have a ton of soil or a ton of dirt from some dig somewhere, and if I could basically grow bacteria in that, I might be able to extract a significant amount of gold. At that point, it might become worth my while. And so it's not just gold, it could be any number of different things you could genetically modify a bacterium to extract from the environment in which it lives. Our last application for genetic engineering is gene therapy. It's the idea that if you've got an individual who has some kind of genetic disease, a disease state caused by a mutated gene or a gene that shouldn't be there, it is conceivable that you could, through this process, actually change the genes in a person to create a situation where we don't have that disease. That is referred to as gene therapy, being able to modify an individual's genes so they don't necessarily have this genetic disease they had once before, and could conceivably improve health quality overall. But I will close in this. Just remember that a lot of these applications that we hear about genetic engineering seem to promise a lot. They seem to be really good. They seem to be really high-minded. We're going to get benefits. We're going to get wealth. We're going to get new products out of this. But just remember that every single genetic engineering project is going to have its potential problems. And sometimes those problems are not able to be anticipated.